Okay, perfect. All right, so in the field session just before this presentation, we had Chris Holcomb presenting on farm water supplies and the evaluation of them. So I'm going to focus mostly on the PMO testing requirements for dairy waters, the actual test procedures that are approved for use to test the different types of water systems, the actual reporting of sample results, and then just tying it all back together. What do those results mean for our processors and our producers, and how are they going to follow up on their water systems based on the sample results that they receive from the labs? So we can go on to the next slide. And just a little outline of what I've broken my presentation out into. Going to start with the importance of water, not just in the dairy industry, but in the food industry as a whole. And I'm mainly going to focus on the dairy industry just because that's my background. And we've got the requirements for testing our dairy waters in the PMO. I'm going to talk about the different types of water that we encounter in the dairy industry, and then the testing requirements and proper reporting for the different types of water, and then our different test methods, because everything's going to be a little different depending on what test method you're using in the lab. I'm going to talk about the approved test procedures for our dairy waters. There's a few different ones that are approved for different types of samples, and we see all of them throughout the labs in New York State. And then how are we going to follow up on unsatisfactory sample results? So our samples that come back positive for total coliform and E. coli, if it's a sample that requires the E. coli follow-up. And then I'm going to bring it back to the laboratory and just touch on the role of the lab, because there's really more to it on the lab end than just getting a sample in and testing it. So we can go to the next slide. So importance of water in the dairy industry or the food industry as a whole. So water is one of the most important and widely used substances in the food industry, and it can be used for a variety of different reasons. Some of them are shown on the slide. So we've got water being used for cleaning equipment and utensils on our dairy farms and in processing plants. We've got animal consumption on dairy farms. Water is going to be used in our sanitization and sterilization processes for your equipment and utensils being used in manufacturing. Water is necessary for hand washing, which is important to prevent contamination of our products. It can be used for heating and cooling products on the farms and during processing. It's used for the production of culinary steam. It can be used to push or convey product during processing, and it can also be used for rinsing our dairy products, such as like the cottage cheese curd. That's one example of where you'll see a product rinse. So water must be from a supply that's properly located and protected. It's got to be easily accessible, and we want it to be safe and sanitary, just to prevent the contamination of our equipment and the product that we're putting out there for consumption of the general public. And just a little note, our polluted water supplies that are used for cleaning utensils and equipment and processing can actually be more dangerous than just consuming the same or similar water supplies of similar quality, because that media that we're working with, that milk, provides the nutrients that bacteria use to grow. So they can grow more rapidly when they're introduced to our processing environment and the food that we're manufacturing for consumption. So a little bit of the different types of water systems and water supplies that we will encounter in the dairy industry. We've got our individual water supplies, and these are going to apply to your farms, your plants, your receiving and transfer stations, your cleaning facilities, and your single service containers and closures plants. And these can consist of your private wells, your springs, any hauled water if you're running short on water and you need to transport water into your plant or onto your farm. That's what we're really thinking of when we think of individual water supplies. These samples, when they're collected and sent in for testing, must be tested for total coliform. And then if your sample results are positive for total coliforms, they're going to require the E. coli confirmation or follow-up on that same sample. How often are we going to test our samples? They need to be collected and tested upon the initial approval of the system. After repairs, modifications, any alterations to that system. And then for our farm samples, they're going to be collected and tested at least once every 3 years. For our plants, receiving stations, and cleaning facilities, those are required to be sampled and tested at least once every 6 months. And then for our single service containers and closures, those are going to be required to be sampled and tested at least once every 12 months. So then going back up to our sample results, if our results are too numerous to count or there's confluent growth, those two terms really apply to our membrane filtration technique, which I'll get to in a little bit. Our sample is going to be invalid and we will need a resample that's going to be tested for heterotrophic plate count. And then if we're talking about our multiple tube fermentation procedure, invalid samples are going to be samples that show turbidity, but they don't show gas production in the conformation stages. And I'm going to talk a little bit about what confluent growth is when we get to that membrane filtration technique. I was able to get some information from the standard methods for examining water and wastewater. So that really explains what we're looking at for too numerous to count and confluent growth. We can go to the next slide. So for reclaimed water, there's going to be a couple different categories that we're talking about when we talk about reclaimed water. So our category 1 reclaimed water is going to be water that's used for potable surf purposes. And this water can be reclaimed from milk and milk products and heat exchangers or compressors in our processing plants. These samples are, or these waters, since they can be used for potable water purposes, are also going to require that E. coli confirmation if our sample tests positive for total coliforms. And again, we're going to use that same sample that tested positive for total coliform to confirm whether or not E. coli is present in that sample. Our category 1 reclaimed water is also going to require a heterotrophic plate count. And I'm going to get into that. I've got a slide just explaining what the heterotrophic plate count procedure is, but the key number we're looking for there is 500. If we have 500 or more colony forming units per milliliter, our sample is going to be positive. And then if it's less than 500, we're going to be not found. So for our testing frequency for our category 1 reclaimed water, for the initial approval of the system, samples need to be collected daily for 2 weeks. And then after any repairs or modifications to the system, they needed to be collected and tested once daily for 1 week. Once we're good, we've got our approval of our system. These are going to require sampling and testing once every 6 months. And then again, if our sample results are too numerous to count, confluent growth, if we have an invalid sample, we're going to have another sample collected and tested for heterotrophic plate count. Next slide. So our category 2 reclaimed water and recirculated cooling water, these are 2 different types of water, but their testing requirements are fairly similar. Our category 2 reclaimed water is not potable and there's limited uses for this water. Some things it can be used for pre-rinsing of product surfaces where pre-rinses will not be used in milk or milk products, and it can be used for cleaning solution makeup water. And there's more uses. Those are just a couple of examples I pulled out of the PMO. Our recirculated cooling water, this is going to be those glycol, sweet water, and those chill water samples. These are going to be used in our plate or tubular coolers or our heat exchangers to cool our product down. These samples are required to be tested for total coliform only, if they're positive for total coliform, we don't need to do that E. coli confirmation. So you don't have to do the testing and you don't report it. Our testing frequency, these are going to be tested initially upon approval of our water system. We're going to test them again after any repairs, modifications, alterations, or disinfections of the supply itself, and then once every 6 months. And then if that category 2 reclaimed water is collected and being stored and we're not doing any treatment on it, then the samples are also going to require a heterotrophic plate counts, and we're going to have to go through that initial approval of the system. So samples for 2 weeks, and then after repairs and modifications, they'll require sampling daily for 1 week. And once again, invalid samples are going to require a heterotrophic plate count on a recheck. Next slide. So our pasteurized equivalent water, this water is typically made with a UV light disinfection process, and it's required for water that comes into contact with pasteurized milk or milk products if that water is not pasteurized itself. And for the initial approval of our system, it's the same as our category 1 reclaimed water. We're going to collect the samples daily for 2 weeks. Following the approval of the initial installation, we're going to sample at least once every 6 months after, and then we're going to do daily testing for 1 week following any repairs or alterations to the system. And this water is actually coming in contact with our pasteurized milk or our milk products. So we are going to require that E. coli confirmation on any sample that tests positive for total coliform. And then the other type of water that we see as our reclaimed farm water. I didn't make a slide for this specifically. The requirements are pretty simple, just like our category 2 reclaimed waters and our recirculated coolants. Samples are going to be collected and tested prior to the initial approval, and then at least once every 6 months. And they're going to be tested for total coliforms only. All right, so next slide, please. So we know what we're testing for, but why are we testing for coliforms? What is their significance and what do they tell us? So first off, what are coliforms exactly? They're facultative, anaerobic, gram-negative, non-spore-forming, rod-shaped bacteria, and they can ferment lactose with production of acid and gas when they're incubated at 35 degrees Celsius. And they can also develop colonies with distinctive characteristics on our endotype media that we use for our membrane filtration technique. Most total coliforms are not pathogenic under normal conditions, but there are some exceptions. So you think E. coli 0157H7, I think we're all pretty familiar with that pathogenic E. coli, which is a coliform. And coliforms are present in the environment and in the feces of warm-blooded animals and humans. So really they can be used to detect contamination of water systems and supplies. And they can be used to indicate that whether or not pathogens could be present in our water system. So overall, they're really just an indicator of water quality. And then together, our test results can be used with results from our sanitary surveys of our water systems to really provide a good assessment of our sanitary quality of water. We don't want to just focus on the physical inspection alone, and we don't want to just focus on our test results alone. We really want to use the information that we get from both of these and bring it all together to really determine whether or not our water system is of a sanitary quality. Next slide, please. So for our approved test methods, a list of these can be found in MA 98. Specifically, Table 4 deals with our dairy waters, so the testing requirements and the test procedures can be found there. And this table will really break down for you which water test methods are approved for which types of water and what our waters actually have to be tested for. So We've got our multiple tube fermentation procedure that can be done as a most probable number or presence absence. We've got our membrane filtration. We've got our chromogenic substrate test methods. So these are your coli-alerts, your coli-sure, your coli-tag, so those types of procedures. And then we've got the heterotrophic plate counts. So I'm going to get into these actual test methods, a little bit about how they work and what water supplies they can be used to test. So next slide, please. So I'm going to start with our multiple tube fermentation. This procedure can be done as presence absence or most probable number, and it can be used on all types of water sources. And then our E. coli confirmation is going to be required for our source waters. So those category 1 reclaimed waters and pasteurized equivalent water as well. And to do this, you're going to be required to use that EC mug broth, and then you're going to have a UV lamp that you're going to use to detect fluorescence. So what do we use for our multiple tube fermentation procedure? We're going to have our double strength lauryl sulfate tryptose broth. So that's your DSLST. And this broth actually contains lactose, which allows for the detection of coliforms based on gas production. So we're looking for that little gas bubble in our Durham tube, which you can see in the top picture shown here. And the sodium lauryl sulfate in this broth is actually going to inhibit the growth of organisms other than our coliforms. So when we see that gas production, we're really going to know that coliforms are present. We're inhibiting the growth of everything else to really just try to narrow down what we're looking for. Our presumptive positive test results are going to show gas production after 24 or 48 hours of incubation. And these are going to be confirmed using the brilliant green bile broth. That's our BGLB. And then we're also going to confirm an EC mug if the sample is one of those water sources that are required to be confirmed for E. coli. If we get turbidity with no gas in our brilliant green bile broth during the confirmation phase, our sample is going to be invalid and we're going to need a resample for heterotrophic plate count. And then if we get that gas production in our BGLB, then the sample is going to be positive for total coliforms. And we're going to look for that fluorescing, which you can see in the bottom picture. That bright blue is what a positive sample will look like, a positive E. coli sample will look like. So how are we going to report our results? This is where most of the errors that I see is in the actual reporting themselves. So if you're using the multiple tubes for the most probable number test procedure, you're either going to have less than 1.1 per 100 milliliters and not found, or greater than or equal to 1.1 per 100 mils and positive. And if we're doing the presence absence procedure, it's going to be less than 1 per 100 milliliters or greater than or equal to 1 per 100 milliliters. And then again, we're going to specify our results as total coliform and E. coli. The E. coli testing is required. So if we have a source water and our results are positive for total coliform, so we have that greater than or equal to 1 per 100 milliliters, we're going to specify that that's total coliform, and then we're going to do our confirmation and report the results for E. coli as well. Next slide, please. So our membrane filtration technique, this one is a little more involved than the multiple tube fermentation procedure. This again can be used on all sources of water, and it's going to use that EC mug and the UV lamp again for our E. coli confirmation. This test procedure can have limitations, especially when we're testing samples that are with a high turbidity, or samples that have large numbers of non-coliform, so like background bacteria. These can make it difficult to read your plates after incubation because there will be a lot of growth on them. And when we're actually performing this procedure, you can see the setup on the top picture. We're going to filter our water sample through a membrane filter, and then those filters are going to be incubated in M-endo broth or on the M-endo agar plates. And after incubation, we're going to read our plates and we're going to decipher what's on them and what needs to be confirmed. So our typical coliform colonies are going to be a red color, and then they're going to have that metallic sheen. It's going to appear kind of a golden green color, which you can see in the bottom picture. It does a really good job of showing us what we're looking for when we're reading these plates. And if you're having a hard time detecting the sheen, the light angle can actually play a role in this. So if you rotate your dish or if you turn it a little bit, you might be able to change that light reflecting on it, and that'll help you discern whether or not we're looking at a typical coliform colony. Our atypical colonies are going to be like a darker red. They might appear mucoid, so kind of viscous and sticky looking. Or we might see nucleated colonies where the center is darker, and these are not going to have a metallic sheen to them. And our non-coliforms, so those background bacteria, are generally going to be a pink color. They might be blue, white, or colorless, and they're not going to have that sheen either. After we read our plates, we're going to confirm in our BGLB, and we're going to use the EC mug as well if it's a sample that needs to be confirmed for E. coli, and we're going to be looking for the same, the gas production in the BGLB and LST. We're going to use the LST too again, and if we've got that gas production, then we're going to use the UV light on the EC mug samples to detect that E. coli if we need to. And then if our samples are confluent growth or too numerous to count, we're gonna have an invalid sample, and we're gonna need a resample collected for the heterotrophic plate count. And standard methods basically says confluent growth is growth covering all or a large portion of our filter, or you can't really see discrete colonies. And then they go on to describe too numerous to count as the total number of bacteria colonies as greater than 200 on any membrane. So if you can't actually get an accurate count, then we're going to need to do that heterotrophic plate count. And for reporting and interpreting these results, we're going to have the less than 1 per 100 milliliters for not found, and then greater than or equal to 1 per 100 milliliters, it's going to be positive. And again, we're going to look for that fluorescence under the UV light if it's a sample that needs to be confirmed for E. coli as well. Next slide, please. So our chromogenic substrate tests, these are pretty common. These can be done as most probable number or presence absence. I don't know that I've ever seen anybody actually doing the most probable number test procedure with multiple tubes. I typically see the one container and just looking for either the presence or absence of the color change, and then the presence or absence of the fluorescing under the UV light. So these test procedures are approved for source waters only. They cannot be used for your recirculated coolants. They can't be used for your category 2 reclaimed water that's not used for potable water purposes. And how they work, they use a hydrolysable chromogenic and fluorogenic substrate to detect enzymes produced by your coliforms and E. coli. And that's what we're actually seeing when we take our samples out of the incubator. The chromogen is going to confirm whether or not coliform are present, and then that fluorogen is what we're looking for the E. coli. And then our different test methods are going to use different substrates to give a different color change depending on which test method you're using when you're looking for that total coliform. So our colilert 18 and the modified colitag are going to turn yellow under the presence of total coliform, and you're actually going to have a comparator that you use to determine your sample results. You got to compare to the comparator and make sure that your results are as yellow or more yellow for that sample to be positive for total coliform. And then your colisure is going to use a different substrate, so we're going to have a red or magenta color if our sample is positive for total coliform. Our E. coli is only approved for presence absence test methods, and we're looking for a blue or a blue-green color change for it to be positive for total coliform. If it's yellow after incubation, we're going to be not found for coliform. And since these test methods are only approved for source waters, any positive result must be confirmed for E. coli because that's the requirement for those types of waters. So we're going to look for that fluorescing underneath the UV light. Reporting for your presence absence is going to be less than 1 per 100 milliliters, not found, or greater than or equal to 1 per 100 milliliters and positive. If you're using the most probable number procedure, it's going to be 1.1 instead of that 1. And then you're going to specify your results again for total coliform and E. coli. Next slide. So our heterotrophic plate count, this is the last test method that we're going to talk about. And this one can be used on all types of water and is going to be required when we're doing a retest on an invalid water system. So any recheck is going to require this heterotrophic plate count. Sorry about the train in the background. So this test procedure is going to be similar to your standard plate count. The main difference is going to be your incubation temperature. We're going to incubate these plates at 35 plus or minus 5 degrees Celsius, just like all of our other water samples, instead of the 32 plus or minus 1 that we use for standard plate count. And this test procedure is actually going to give us an estimated number of live culturable heterotrophic bacteria in our water supply, in our water sample. For our interpretation, like I said earlier, we're looking for that 500 number. So if we're not found, we're going to be less than 500 CFUs or colony forming units per milliliter. And our positive samples are going to be those that are greater than or equal to 500 colony forming units per milliliter. Next slide, please. So interpreting results, what does this all mean? We get our results, we report them, we send them back to our processing plant, our producer, whoever sent the sample in, and now they need to try to figure out what these results mean and what they're going to have to do for a follow-up on their system. So if our water sample was not found for total coliform, we've got a satisfactory system, so everything is good, and we'll just continue sampling at the next required frequency. If our sample is positive for total coliform only, that system is going to be considered at risk, and the processor, the facility, is going to have to do some follow-up. If we're positive for total coliform and E. coli, we have an unsatisfactory system, and again, we're going to have to follow up on this, but this time the regulatory agency is going to get involved, and they're going to be involved in that follow-up, and I'll explain what the follow-up is in a minute. And then our 2 numbers to count are our confluent growth samples. Those are those invalid samples. We're going to follow up with a heterotrophic plate count, and if our plate count is less than 500 milliliters, we're satisfactory. If we're greater than or equal to 500 CFUs per milliliter, then we're unsatisfactory. Our sample is positive, and that's where we get that at risk from. Can you go to the next one, please? So following up on an at-risk water system, again, these are going to be our systems that are positive for total coliform, not found for E. coli, or if our heterotrophic plate count is greater than or equal to 500 CFUs per milliliter on a previously invalidated sample. And for following up, the individual facility or the processor is going to be required to complete a physical inspection of their water system within 30 days of that positive sample result. So that 30 days is not from the day they get the result, it's from the day that that system actually tests positive. And if they do not complete this inspection or they don't complete it on time, then their system is now going to be considered unsatisfactory, and the regulatory agency is going to step in and do that physical inspection. So after the physical inspection is done, the facility is going to be required to make any necessary corrections, and a resample is going to be taken and tested, and hopefully that comes back satisfactory. If it's still testing positive, then the regulatory agency comes in and conducts an inspection, and you just kind of repeat the same process. Next, please. So for our unsatisfactory systems, again, these are systems that samples test positive for total coliform and E. coli, or if that water system inspection was not completed within 30 days. They're gonna require a physical inspection from the regulatory agency. The facility is gonna have to make any and all necessary corrections, and another sample is gonna be taken and tested for bacteria, and hopefully that sample is satisfactory. If it's not, then again, continue with the inspection and correction procedure until we get a satisfactory sample. So then one more slide, next one please. So tying it all back to the laboratory now, what is the lab's actual role? Other than actually just doing the testing themselves, they're going to be responsible for receiving samples, or, you know, more importantly, rejecting samples that do not meet the requirements of the water sample itself. So samples must be transported and maintained at 0 to 4.5 degrees Celsius. If the samples come in unrefrigerated, they're going to be rejected if transit time exceeds 6 hours. Total transit time needs to be less than or equal to 30 hours. And you need a total of 100 milliliters for testing. So if a sample comes in underfilled, we can't do the testing on that because there's not a sufficient volume. Those have to be rejected. And then if they're overfilled to the point where there's not enough airspace in the sample container for agitation or mixing the sample, then those will have to be rejected as well. Testing must be completed within 30 hours of collection or within 2 hours of receipt of those samples at the lab. And the lab must use an approved test method to test those samples. So just some things to think about. The chromogenic substrate test method is only approved for source waters, so you can't use that for your coolants that come in. And then if you get a tinted water sample, that color can interfere with the color change you're looking for during the testing for that colilert and colisure. Sometimes samples come in with like a yellow tint to them, and that could interfere with trying to determine whether or not your sample changed to yellow. Or I've seen notes on the bottom of water sample reports where the sample was tinted. It must have been like a rust tint, and there was like a red color to the sample. Those might interfere with your colisure testing because we're looking for that red or magenta. So you might need to use a different test procedure on those samples. Your results need to be reported at each step of incubation, and they need to be reported and interpreted correctly. So make sure you've got that 1.1 per 100 milliliters for your most probable number versus your presence absence of 1 per 100 milliliter. And make sure your reporting is positive or not found for total coliform and positive or not found for E. coli if that confirmation is required. Do not test recirculated or category 2 reclaimed water for E. coli. That's right in that MA98 table 4. And if you don't do E. coli testing, don't record results as not found. E. coli confirmation must be completed when required. And then I think probably the most important thing to remember is prompt notification, especially when our results are unsatisfactory, because that follow-up on the facility needs to be done within 30 days of that positive test result. So if you're late in getting those results out, you're cutting into the amount of time that facility has to complete their follow-up inspection and corrections and get that sample, which is required in the PMO. So that's really all I've got. If you go to the next page, I've got my contact information here. If you have any questions or need anything, whenever I'm looking stuff up for dairy waters, I go to the Pasteurized Milk Ordinance, and I'm sure everybody probably already knows by now that the 2019 revision has been released. If you want a copy of that and don't have one already, you can find that on FDA's website. Our Dairy Waters 2400 form, which is the form we use when inspecting our labs that do dairy water testing. That was currently revised in October, and I think that was actually sent out earlier this spring. If you need a copy of that, that too can be found on FDA's website, or I can send you a copy of that. MA 98, again, the current revision is April 2020. And then that standard methods for the examination of water and wastewater is a really great resource to have available as well.
I put links to the majority of those in the chat box too for everybody.
Thank you for doing that, Barb.